Namaskar, welcome to PK's classes. Today we will study mechanism of resistance to anti-malarial drugs. So before discussing the mechanism of resistance, let us first discuss what is malaria and what are the anti-malarial drugs. Then we will discuss the mechanism of action of those anti-malarial drugs and finally mechanism of resistance. So malarial parasite is known as the Plasmodium, and there are 4 species: Plasmodium vivax, Plasmodium falciparum, Plasmodium ovale and Plasmodium malariae. And the most common species are vivax and falciparum. And this Plasmodium requires 2 hosts. One is the human beings and the other is the female Anopheles mosquito. And accordingly, it has 2 cycles. One is the life cycle can be divided into asexual life cycle and the sexual life cycle. And the asexual life cycle occurs in the human beings, whereas the sexual life cycle occurs in the mosquito gut. So the gut of the female Anopheles mosquito. So when a mosquito bites, it releases sporozoites. These sporozoites enter into the liver, and in the liver the sporozoites are processed to merozoites, and finally the merozoites are released. These merozoites attack the blood cells, that is the erythrocytes, and some of the merozoites, while entering into the RBC, are processed to form the male and female gametes, and these gametes are taken up by the mosquito, and inside its gut they are processed. So gametes form the zygote. Zygote is converted to ookinete. Ookinete is converted to oocyst, and finally oocyst is converted to the sporozoites, which are again transferred to another human being to which the mosquito bites, and in this way this life cycle continues. And the merozoites, after entering into the blood cells, that is the erythrocytes, again the erythrocytes release the merozoites, which can attack the fresh hepatocytes or the liver cells, and that stage is called exoerythrocytic stage. So the asexual cycle can be divided into pre-erythrocytic stage, that is before entering the blood cells, the RBC. So from sporozoites to these merozoites, that is the pre-erythrocytic stage. Then attacks the RBC, that is the erythrocytic stage. And some of the merozoites can attack the fresh liver cell again, and they can be stored there as the dormant forms, hypnozoites, and that is called exoerythrocytic stage. And exoerythrocytic stage is absent in Plasmodium falciparum and Plasmodium malariae. So just see, so this is pre-erythrocytic stage, and this is erythrocytic stage, and some of the merozoites can again attack the fresh liver cells, that is exoerythrocytic stage. So the in the erythrocytic stage, the RBC is attacked by the merozoites, and initially there will be ring stage by 12 hours, ring-like structure, ring stage, then early trophozoite stage, then late trophozoite stage, and finally the scission stage. By 48 hours it bursts to release again merozoites. So the drugs which act in this erythrocytic stage are called blood seasonicides, and the drugs which act in the liver, kills the hypnozoites, they are called as tissue seasonicides. And some of the merozoites in the initial stages can be converted to the male and female gametes, and these some of the drugs they can act on these gametes. They are called gametocytes. And some of the drugs, they inhibit the process of gametes to sporozoites in the mosquito gut, and they are called as the sporondicides. And some of the drugs can inhibit this link between the pre-erythrocytic stage and the erythrocytic stage by killing these merozoites. They are called as the chemoprophylactics. So the blood seasonicides can be of two types: low acting and low efficacy, fast acting and high efficacy. The fast acting and high efficacy are of two types, which act on the signet ring stage and early tophozoite. They include the artemisinin and its derivatives like artemether, arteether. And the second group, they act on the late tophozoite and scission stage. They include the 4-aminoquinolines, aryl amino alcohols like quinine, and naphthoquinones like atovaquone. And the second group, the slow acting and low efficacy drugs, include the antibiotics like doxycycline, clindamycin, and the folate synthesis inhibitors, the sulfonamides, proguanil, pyrimethamine, cycloguanil, etc. So the third group of drugs, that is the tissue seasonicides, they mainly act on the hypnozoites, and they include 8 aminoquinolines like primaquine, tafenoquine, bulaquine, etaquine, and they act on the exoerythrocytic stage. So they are called as secondary tissue seasonicides, and those which act on the primary pre-erythrocytic stage, they are called the primary tissue seasonicides. They include the dihydrofolate reductase inhibitors like proguanil, pyrimethamine. So tissue seasonicides can be either primary or secondary. Primary act on the pre-erythrocytic stage merozoites, and secondary act on the exoerythrocytic stage, the hypnozoites. Besides them, there are gametocides like primaquine, chloroquine, quinine, and the new group of drugs, pyroindolones, and sporontocides like primaquine, proguanil, pyrimethamine. They act in the mosquito gut and prevent the formation of sporozoites from the gametes. Then chemoprophylactics, they kill the merozoites released from the liver, and they inhibit the link between the pre-erythrocytic stage and erythrocytic stage. They include chloroquine, mefloquine, proguanil, doxycycline, and clindamycin. Besides the desferrioxamine, the iron chelator can also be used, which binds to iron and form complex, deprive the parasite of iron, the vital nutrient, and inhibit the growth and survival of the parasite. Albetiazolium is another new drug which inhibits choline uptake into the parasite and inhibit the synthesis of phosphatidylcholine, a key component of cell membrane, and cause parasite death. Besides inhibiting choline uptake, it also inhibits certain enzymes required for the synthesis of phosphatidylcholine. And another new drug from the Chinese herb Changshan, Dicroa febrifugin, and its derivative halofuginone. They inhibit the hemojoin formation, inhibit protein synthesis by inhibiting propyl tRNA synthetase, and also increase the nitric oxide production. So the molecular targets of antimalarial drugs can be divided into heme detoxification in the digestive vacuole or food vacuole, folate and pyrimidine biosynthesis in the cytosol, electron transport in the mitochondrion, and protein synthesis. Particularly this is in the apicoplast. So first let us discuss the first one, the heme detoxification. So the malarial parasite, this is the malarial parasite Plasmodium, which is present inside a parasitophorous vacuole, which is inside the RBC. So the malarial parasite enters the RBC because RBC contains hemoglobin, which acts as a food, and the malarial parasite has a food vacuole or digestive vacuole, and hemoglobin enters from the RBC into the food vacuole. And this hemoglobin by parasitic proteases is divided into heme and globin. Heme is the ferrous form, which is toxic to the parasite, and heme is converted to hematine, the ferric form, and then dimerized by the enzyme heme polymerase to hemojoin, and hemojoin is non-toxic or neutral to the parasite. So the heme polymerase is released by the malarial parasite, and drugs like 4-aminoquinolines, chloroquine, they inhibit heme polymerase so that there will be accumulation of toxic heme and the malarial parasite dies. And globin is used as food by the parasite, and globin is degraded by the proteinases, the plasmodium aspartic proteinase, the falciparum cysteine proteinase, which convert globin into 10 to 20 amino acid oligopeptides. Then they are further degraded to 5 to 10 amino acid oligopeptides, then into dipeptides and small peptides, and finally amino acids. And these amino acids are used by the malarial parasite for the synthesis of proteins. So the artemisinin alkylates the heme, the ferriprotoporphyrin 9, the heme, and form the hematine artemisinin adduct, and this hematine artemisinin adduct inhibits heme polymerase and causes accumulation of toxic heme, which kills the parasite. And it also cleaves and activates the peroxide bridge present in the artemisinin structure, and generates highly reactive free radicals, mainly the carbon-centered free radicals, which bind covalently to the parasite membrane proteins and lipids and damage the parasite membrane. Then we'll go to the electron transport chain. Electron transport chain, we know, is required for the synthesis of ATP, and the electron transport chain is consisting of the dehydrogenases, the complex 2, the coenzyme Q, then the complex 3, the cytochrome BC1 complex, complex 4, and ATP synthase enzyme. And the electron transport chain provides electrons for the coenzyme Q or ubiquinone dependent this enzyme dihydrofolate dehydrogenase, because this enzyme is required for the synthesis of pyrimidines. So dihydrofolate to orotate, then orotate to pyrimidine. So two functions are there of electron transport chain. One is ATP synthesis, and the second is the pyrimidine synthesis. And the drug which acts on the electron transport chain is the atobacone, and this can inhibit ATP synthesis. It can also inhibit the pyrimidine synthesis. Then this is the dihydrofolate to orotate, and here the type 2 dihydrofolate dehydrogenase enzyme, and here we use the ubiquinol or coenzyme QH2. It is converted to coenzyme Q or ubiquinone, and this regeneration occurs by the complex 3 cytochrome BC1 complex, and atobacone inhibits the cytochrome BC1 complex. Then the production of tetrahydrofolate by the malarial parasite from guanosine triphosphate GTP, and here the key enzymes are dihydropteroate synthetase and the dihydrofolate reductase. So the sulfonamides inhibit DHPS, and the drugs like cycloguanyl, proguanyl, and pyrimethamine inhibit dihydrofolate reductase. And tetrahydrofolate actually acts as a coenzyme to the enzyme thymidylate synthetase, which converts deoxyuridine monophosphate to deoxythymidine monophosphate, which is required for DNA synthesis. Then the Plasmodium, the malarial parasite, has another organelle, that is epicoplast. The epicoplast is a four-layered non-photosynthetic plastid, which has its own ribosomes, and these ribosomes, by using that, they synthesize the proteins required for its survival. Besides that, the apicoplast also has functions in fatty acid synthesis, isoprenoid synthesis, heme biosynthesis. So the antibiotics like doxycycline, clindamycin, they inhibit the protein synthesis by binding to the ribosomes present in the apicoplast. So the malarial parasite has its own ribosomes, and besides that it has a single mitochondrion where the electron transport chain has its function, and there is apicoplast where the apicoplast also has its own ribosomes. So now we'll discuss the mechanism of resistance to antimalarial drugs. So the first mechanism can be the alteration of the drug target, so that the affinity of the drug for the target will decrease. For example, mutations in dihydropteroate synthetase causes resistance to sulfadoxine. Mutations in dihydrofolate reductase causes resistance to pyrimethamine and cycloguanil. And single point mutation in cytochrome B subunit of the BC1 complex, that is complex 3, atophycon resistance results. Point mutations in the apicoplast encoded 23S RNA, so doxycycline and clindamycin. Then drugs which can for which resistance can be produced by increasing the efflux of the drug. The malarial parasite increases the efflux of the drug and decreases the drug concentration in the food vacuole, and this is done by the mutation of two genes. One is the PFCRT gene, that is Plasmodium falciparum chloroquine resistance transporter gene, and here resistance to drugs like chloroquine occurs by single point mutation K76T. And there can be mutation of Plasmodium falciparum multi-drug resistance 1 gene, PFMDR1 gene, and for example mefloquine mutation, mefloquine resistance occurs here. So you can see here in this figure, these are the efflux pumps, and there can be these two enzymes which mutation can result in this, and then again resistance to atophycon can be caused by mutation in the cytochrome B subunit of the BC1 complex or complex 3. Then next is the third mechanism is parasite tolerance to oxidative stress. There can be alterations in the parasite redox system, upregulation of antioxidant mechanism like glutathione, peroxyredoxins. So they will detoxify the reactive oxygen species. So the mechanism of primaquine is inhibited because primaquine is a prodrug, and its quinone metabolite inhibits the coenzyme Q governed respiratory process of the parasite in the exoerythrocytic stage and kills the hypnozoites. So primaquine is the prodrug, and its quinone derivative actually causes the death of the parasite or the death of the hypnozoites, the killing of the hypnozoites by a coenzyme Q governed respiratory process inhibition. So that requires the reactive oxygen species, and those can be detoxified by the antioxidant mechanisms. So this is another mechanism of resistance. Then the fourth mechanism of resistance is the mutations in the K13 gene, that is the KL13 propeller domain in malarial parasite Plasmodium falciparum. And let us see this figure. So this K13 is required for the uptake of hemoglobin. So as we have already discussed, that hemoglobin has to enter from the RBC into the food vacuole, and that is required by this protein, this KL13 propeller domain, and this protein is encoded by the gene K13 gene. And when there is mutation in the K13 gene, there will be no uptake of hemoglobin from the host RBC. So there will be delay in digestion of hemoglobin by the parasite, and that causes the partial loss of function and decrease in the effectiveness of artemisinin, because artemisinin requires heme for its activation, and because artemisinin alkylates the heme, and there will be formation of artemisinin and hematin adduct. So that activation of artemisinin is not possible, so that will cause resistance to artemisinin. Next mechanism is the increased expression of hemoglobinases. For example, as we have already discussed, that the hemoglobin is converted to heme and globin. Globin is the food, and this globin is processed into amino acids by the enzymes, and if there will be increased expression of these enzymes, then it will provide the parasite with amino acids and will facilitate the protein synthesis. So it will help in the growth of the malarial parasite. So that can be also a cause of the resistance of the malarial parasite, resistance of the anti-malarial drugs to the malarial parasite. Then finally, the sixth mechanism is reduced drug activation by changes in parasite enzymes. So sometimes there can be polymorphisms in the cytochrome P450 group of enzymes like CYP2D6 in the host, and that causes the decreased activation of certain drugs like primaquine. As we have discussed, that primaquine is a prodrug. It is metabolized to its active quinone form, and that is decreased or reduced if there is polymorphisms in the CYP2D6 group of enzymes. So that decreases the efficacy and causes the resistance. So this is all about the resistance to antimalarial drugs. Thank you.